Welcome everybody to the 85th Forest Industry Lecture Series. So my name is Brad Pinno. I'm Assistant Professor of Silviculture in the Department of Renewable Resources, and I will be the Master of Ceremonies here today for our event. So normally at Phil's we have a speaker coming in, a nice lecture hall, reception afterwards, and obviously we can't do those sorts of things now, but I think what we've got for you all today is really exciting. We're able to focus on all of the great research that's happening with our graduate students and postdocs in our department. So it's given us an opportunity to highlight the work that's happening at the University of Alberta related to forests and forestry. So please know that the University of Alberta acknowledges that we are located on Treaty 6 territory and respects the histories, languages, and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. So the program that we have today, we've got it split up into a few different parts. We have a quick introduction here. We're going to have a poster session. We're able to move around to different tables, see different posters from our graduate students. Then at about 3:45, we're going to have our presentation. So we have 3 presentations from postdocs in our department. And then after that, we're going to have another short poster session. And then we'll wrap up around 5 o'clock this afternoon. So before we get started, I just wanted to take a minute to thank our sponsors. So without our sponsors, this event just doesn't happen. And I think this is really a testament to this being an important event and well supported by everybody in the industry here in Alberta. But along the bottom of your screen in Remo there, you can see the list of all the sponsors. You can see all of their logos and such. But I just want to take a second here and read out these sponsors. So if you're sitting at a table with anybody from these organizations, give them a big thank you, okay? So in alphabetical order, the sponsors for the Forest Industry Lecture Series are Alberta Agriculture and Forestry, Alberta Forest Products Association, Alberta Pacific Forest Industries, ANC Timber, Association of Alberta Forest Management Professionals, Canadian Forest Products, Canadian Institute of Forestry, Rocky Mountain Section, Carson Integrated, DLA Piper, 4Corp Solutions, Foresight Consultants, FRI Research, GeoVera, Greenlink Forestry, Incremental Forest Technologies, Miller Western Forest Products, MNP LLP, Natural Resources Canada Northern Forestry Centre, Norbord, Tolko Industries, Vanderwell Contractors, West Fraser Mills, Wild and Pine, and Woodlands Operation Learning Foundation. So a big thank you to all of our sponsors. I'll have some more information on kind of the logistics of what we're going to do for our poster session afterwards. But for right now, I want to turn it over to Ellen McDonald, who will bring greetings and updates from the faculty and department.
Okay, thank you, Brad, and welcome everyone. It's my great pleasure to be here. I'm Ellen McDonald, Chair of the Department of Renewable Resources, and I'm very, very pleased to welcome you all to our 85th Phil's Lecture on this beautiful Thursday afternoon. It's really wonderful to connect with you all, even remotely. So far, I'm pretty excited about Remo, and I'm optimistic that it's going to run smoothly. Our Dean Stan Blade is with us here, but for some very strange and frustrating technical difficulties, he's not able to get up on the stage and speak. So you can circulate and say hi to him later, and I do want to say he sends his very warm greetings and has passed on his messages to me that he wanted me to share with you. If you're like me, these days in March now are bringing back very vivid memories from a year ago and the rapid way that the world was changing at that time as COVID became a reality. And Phil's actually last March 5th was our very last public event prior to the lockdown. So it was on March 5th, and 8 days later the university was closed. Yeah, so that puts in context how long this has been going on, and we are all very much looking forward to return to circumstances where we can meet in person. I really want to thank the Phil's team for pulling this all together, Brad Pinno, Barb Thomas, and Marty Luckert. with support from Rahana Bennett, Christina Host, Cynthia Strawson, and Byron Flores. Really a lot of work to put this together, and I'm optimistic it's going to run very smoothly. And I want to thank the team in particular for coming up with this very inspired choice of selecting current and future forestry research leaders to be the focus of this year's lecture. It seems like it was just yesterday or possibly 5 years ago that we had our very fun fills in December, but I do have a few things to report on since then. So the winter term has run quite smoothly. We had a similar model as in the fall, so we have a very few in-person classes, the forest health class, courses in moss and lichen identification, some soils courses, a few field trips we were managing to have in person, and everything else was online. The university has stated that they plan to move to more in-person classes this fall, and we're in the process of planning what that looks like, looking at available classroom space, presuming that some physical distancing will be required, and how many students we can fit in, and which courses should be the priority to have for in-person. So we'll be clarifying that in the next 2 or 3 months as we go forward. Our usual spring field school, we decided to have it again in August this year, which is what we did last year. So a 10-day intensive in August with daily trips from campus, because I'm quite certain nobody's going to let us have students sharing accommodation and food in May this year. And the fall field school for first-year forestry students that Brad Pinno runs is also likely going to run in that same way with daily trips from campus, and like he did this past fall, be augmented by a series of 1-hour follow-up sessions during the fall term so that students can connect better with professors and with professionals and with one another. So I think we've done a great job, and I want to thank all our faculty and staff for their tremendous efforts to continue to deliver a really quality learning experience to our students through these really difficult times. Another happy thing that I can share with you is I'm very pleased to announce that Dr. Robert Froese, who is currently at Michigan Technological University, has accepted the position of the Endowed Chair in Forest Growth and Yield. In our department, he's going to officially join us on June 1st, but he's already getting quite engaged in activities in Alberta, and we really look forward to warmly welcoming him to our ranks and all the great works he's going to do for our programs and for Alberta's forests. I want to again express my very heartfelt appreciation to the donors who made it possible to create this endowed chair. So Robert is actually with us here today, but of course, I don't know, he can wave from his table. It's not actually possible. He's here today, and if you go to the first floor in Remo, you will see a table that has his name marked on it. So he's going to be hanging out at that table during the first poster viewing time if you want to stop by and say hi to him and meet him. I do have some sad news to share, which many of you may have already heard, which is that Dr. James Beck passed away on February 16th. So as many of you know, Jim Beck was a graduate of Berkeley and one of the first 2 professors hired to start the forestry program at the U of A in 1971. He went on to become chair of the Department of Forest Sciences in 1983 to 89 and chair of the Department of Renewable Resources when we merged and changed names, and that was from 1995 to 2001. And he was an amazing champion of our department, our programs, and he was very deeply influential in the forestry community and also the natural history communities in Alberta through his interests in birding and butterflies. And anyone that's been at a Phil's in the past knows that his booming voice calling attendees to move to dinner from the reception at Phil's is historic. So Jim will be very deeply missed by his wife Barb, their 2 sons and their families, and his many, many friends and colleagues at the University of Alberta and throughout the province. The university flag is being lowered in his honor from yesterday through tomorrow, the 3rd to the 5th, and we aligned that so that it would bracket the Phil's lecture today. In terms of our programs, over the last 1.5 years, we've been working on a major in-depth review of our BSc in Environmental and Conservation Sciences and the majors, and we're at the stages now of implementing recommendations from that review by going forward with calendar changes. Because the NCS and forestry programs have close alignment, this is going to precipitate a few minor changes to the forestry program. One of the most important is going to be the addition of a required course on Indigenous knowledge and Indigenous perspectives on environmental issues. I'm very excited about this. I think this is a very forward-looking step towards addressing the recommendations of the Truth and Reconciliation Commission and providing our students with some very much needed content and understanding. So that will be coming, I believe it will be coming part of a required course in the forestry program moving forward. As you may have heard, the university is going through some major changes, academic and administrative reorganization, which were precipitated by budget cuts. One major change is that faculties are now going to be grouped into colleges. So some of the important administrative and academic management and functions will occur at the level of the college rather than the faculty. So as of July 1st, our faculty, Agricultural Life and Environmental Sciences, will be combined with the Faculty of Science and the Faculty of Engineering into the College of Natural and Applied Scientists. We're actually optimistic this is going to provide increased opportunities for cooperation and collaboration with those faculties. And in fact, we've already implemented a coordinated effort with Faculty of Science for student recruitment, and I think that's a very good thing. We anticipate it's going to lead to increased enrollment because it's going to help students find our programs. So if they go to the Faculty of Science first because they go, oh, I want to do science, then they'll be presented with an array of programs, including the programs in our faculty. The university has pretty ambitious plans to grow enrollment, and we hope to be an important part of achieving that. I will just end by noting that this is going to be my last fills as the department chair. My term is ending on June 30th this year, and I'm not standing for another term. The dean has begun the process to select the next department chair, and I'm confident that the department will be in very good hands, and you will meet whoever that new chair is at fills in the fall. That's all I have. Again, thanks everyone for coming. I welcome you. Enjoy the rest of the event, and I'm going to turn it back over to Brad now to explain how all this is going to work. So thanks, Brad.
All right, thank you, Ellen. So now I'm going to go through some of the kind of the operational details of what we're going to be doing for the next little while here in regards to the poster presentation in particular. So we have 20 posters from graduate students today that are being presented. So each table has a graduate student at it, or not all of them, but most of the tables have a graduate student at them with, they'll have their name as well as a general topic for the for their poster. So you can see that. You can go and visit them to talk with the students, learn a little bit about their project. Okay, so when you get to the table, you can turn on your mic and your camera so you can actually see and talk with folks. You can see the names of the participants in the whole event by hovering over their the letter icon. You can see what their actual name is. So if there's people you want to talk to, or perhaps people you want to avoid, you can do that. There are posters located on 3 floors. So we have them on floor 1, floor 2, and floor 3. Okay. So you can see there's an elevator on the left-hand side of your screen. You can select the floor you want to go to, and you'll go up or down regardless, or to that floor. Okay. But there are posters on all different floors. So to move around tables, you may have been experimenting with this already a little bit, but you click on a blue chair icon and you can move from table to table to table. So I'm just looking at the number of attendees we have. We have just about 150 people here today, and we have 20 posters. So there's going to be a fair amount of people at each poster. So please kind of keep moving through them and try and get to a few of the different posters here in our first session, because other people will probably be waiting to get to see those posters as well. If you need a list of the poster titles and presenters, that's in the agenda, and I think it's been posted. Yeah, it's been posted in the chat box there now, so you can see that if you want any more details about them. If there's any posters in particular you want to make sure you get to. A couple other things. There are a couple of networking and open tables. If you want to just sit and have a conversation with somebody, those tables are available. But there's no posters of those ones, obviously. We also have a help table. If you need help with anything, there's a help table on the first floor. It's labeled help. You can go there, and there's people there that can help you, navigate the system here and get to where you need to go and how you need to get there. A couple of things on just using this, the buttons for turning your camera on and your microphone on at the bottom of the screen. If there's something you need blown up or larger on your screen, you can click on it and it will expand it, like the posters, for example. If you click on the poster, it will expand it larger, so you should be able to see it a little bit better if you need to. The other one, like Ellen mentioned, Robert Froese has a table here on the first floor if you want to talk with him, our new growth and yield chair. He's on the first floor. He'll be there for this first poster presentation, but I think during the second one he'll want to see some of the posters too. So if you want to talk with him, now is the time to do that. Last thing I want to mention before we send you off into the world of posters is we do have poster prizes available for the top 3 posters to be won today, but we need your help, okay? So we're going to be posting in the chat box a link to a Google Doc. There it is. Thanks, Rohan, for posting that. Please, once you've gone through a few posters, click on that Google Doc, select the posters that you would like to vote for these prizes, okay? And we'll announce them at the end of the night tonight, okay? So that's kind of some logistics. What we've got is we've got the next 25 minutes for a poster session. We'll all reconvene here at 3:45 for the presentation from our postdocs, and then we'll have another poster session after those presentations. So don't worry if you're not able to get to all the posters you want to now. You will be able to do that in the second half of the poster session after the presentation. So with that, please remember posters on floors 1, 2, and 3. Cycle through the different posters and see as many as you can, and we'll see you back here in about 25 minutes. Enjoy, everybody. All right, well, welcome back, everybody. Hopefully everybody got a chance to have a look at a few posters. I understand that it was pretty rushed, and I only got a chance myself to go to 4 posters, but hopefully some of you are able to go through a few more and talk to a few people, which is what this is really all about. So now we're going to move on to our next part, which is the presentation. So we have 3 presentations from postdocs who are in our department here. And I guess the other thing I'll just mention is we are going to have another poster presentation or another poster session after our presentations. So you'll be able to go back to any of the other ones that you didn't get a chance to visit in the first go round. Okay, but let's just jump right into the presentations here. So our first presentation is from Esteban Galeano, who is a postdoc in the tree improvement lab with Dr. Barb Thomas. And he's going to be talking about implementing cutting-edge molecular technologies in Alberta's tree breeding programs to increase genetic gain. And maybe just before we start the video, one thing I will mention is what we're going to have time for 1 or 2 questions after the presentation. If you have any questions, please put them in the chat box. I'll be monitoring that, and then we can ask those questions after the presentations here. So if you have questions, into the chat box, please. And with that, let's have our first presentation.
Hello, everyone. My name is Esteban Galeano, and I work with Barb Thomas at the Department of Renewable Resources. Today, I will be talking about implementing cutting-edge molecular technologies in Alberta's tree breeding programs to increase genetic gain. So first of all, let's introduce the concept of tree breeding cycle. So how it works. First, we select parents from natural stands to establish the base population. After selecting, we perform breeding to establish the breeding population, and we test the families from the breeding population in progeny trials, and after testing, we select the best families and the best parents to establish the production population, which is called seed orchards. After the production, we can continue our cycle for a 2nd or 3rd generation orchards, and the goal is to maintain a balance between the genetic gain and to ensure diversity. I would like also to introduce the concept of pollen contamination. So let's imagine that we have our orchard in here, our seed orchard or the population, and we have in here a natural stand or another orchard from the same species, and unfortunately we can get contamination from pollen out of our orchard. This is a potential problem for most orchards around the world, and this can potentially decrease the genetic gain. Also, let's introduce the concept of genetic gain. It is obtained by selection, but it's affected by pollen contamination. So let's imagine a distribution of phenotypes in the parental generation with small trees in the population, with average trees, and with big trees. And these ones are the ones that will be selected as parents. In the next generation, we will have offspring produced from these parents, and they will configure a new distribution of phenotypes. We will have the phenotype from parents, which is corresponding to this average, and we will have a new population, which is the offsprings, with a better performance. The difference between the offspring and the average of the parents is called genetic gain. And the genetic gain is calculated by heritability multiplied by selection differential, which is what I explained, the difference between the average of the offsprings and the average of the population of the parents. And the genetic gain needs to be multiplied by the pollen contamination level. And this is how it's so important to have accurate pollen contamination levels. Because if we have 50% of pollen contamination, we can decrease half our genetic gain. And that's why we have been fostering the idea of genotyping different breeding programs in Alberta. Genotyping is the same as saying paternity testing, and how it works. Let's imagine that we have 8 trees that we want to genotype, and we want to know the relationship between them. So we collect the needles, we extract the DNA, and we will be using this SNP chip to understand the DNA and to understand the relatedness between them. The SNP chip will receive in each cell the DNA, and it is going to be placed in this equipment from Illumina company. After this, we will get this kind of diagrams called dendrograms to understand the grouping or the relationship between them. In here, you can see that tree 1, 2, and 3 are close related. Tree 6, 7, and 8 are close related, but tree number 5, it is not so related with the rest of them. This information can be used to measure diversity, calculating effective population sizes, and contributions of male and females. And also, we can use this information to increase gain by reconstructing pedigrees and measuring pollen contamination levels. So I would like to bring a case study, which is one of the white spruce breeding programs that we have been analyzing. It is called G1. We collected needles from founders close to Smoky Lake, seedlots from seed orchard in Grand Prairie, and progeny trials. And we asked these questions. How is the diversity in the different populations? How are the male and female contributions in the seedlots? And how is the pollen contamination, and how this can affect the genetic gain? So we went to the founders, and we collected the needles using pole pruners, and we went to the progeny trials, which have smaller trees, and we used scissors, and we bagged all the samples, and we kept them cold. So let's see some results. First of all, in here I am showing in blue columns the diversity, which is expressed in effective population size, and in the red dots I am showing the relatedness. The highest diversity was found in founders, with the lowest relatedness, which is what we desire. And for seedlots 2003 and 9, we found a decrease in diversity and a slightly increase in relatedness. But after eliminating 65% of the trees in the orchard, and this elimination was based on performance from height of genotypes, we found a significant increase in relatedness and a significant decrease in diversity. The same with contributions. For 2003 and 2009 seedlots, we have balanced contributions from mothers and fathers, but after elimination of those genotypes, we found an unbalanced contribution. Let's see pollen contamination. What we found, different levels of pollen contamination from seedlots going from 50% down to 10%, and they correlated exponentially with wind direction. This correlation helped us to understand what was going on in the site. In these black squares, we can find other seed orchards that are not white spruce, and the white spruce ones are in here, and we found that the contamination source, it is located in here, and we have a significant external pollen coming from the target orchard of study. Then we could reconstruct pedigrees with this genotyping technology, and don't panic with this graphic. I know that it's quite complex, but this is showing all the structure of the population with the females in colored dots, the males in black dots, and we can understand how the families are configured. And also we can find and understand the pollen contamination in seedlots and in progeny trials. This information can be used not only to find the pollen contamination, but also to get better estimates of genetic gains. So as conclusions, as our recommendations, first, the usual recommended distances between orchards from the same species or orchards and natural stands of 1 kilometer are insufficient to ensure low contamination levels. For this, we recommend to use drones carrying pollen. They are called dropcopters. Second, random mating does not happen in operational tree breeding, so a careful design of breeding programs is needed. For this, we have different software, and I am just bringing an example as PopSync. Third, tree breeders should avoid severe ruggings to prevent increases in relatedness and genetic diversity losses. One solution could be to collect pollen from the best males and pollinate the best females to get improved seed. And finally, we definitely recommend the adoption of genomic tools to advance the tree breeding programs in Alberta using SNP chips. So the perspectives for the different Alberta programs are the use of genotyping for accurate pollen contamination assessment, progeny testing and selection of parents, accurate effective population size calculations, understanding of contributions from male and females in seedlots, the first two ones to increase gain, and the last two ones to ensure diversity to follow the public policies in Alberta, and to have a balance between gain and diversity. I would like to thank our lab sponsors, all the team that have been helping us through all these years, and of course the field sponsors. Thank you very much again, and I am open to questions.
Okay, thank you, Esteban.
Thanks.
So one question we got was regarding the drop copters. Can you expand on that idea a little bit? That seemed pretty interesting. The idea was you were going to use them to actually aid in the pollination in the orchards. Is that correct?
Yeah, I think that Barb has been bringing in the last year. It is really recent, this idea. It has been done in other species in the US. and it seems to be a very promising way of like blowing a certain amount of pollen in the orchards, and then this is like a very nice idea to do it. But we have other ideas to avoid like pollen contamination, like supplementary mass pollination and other strategies, but this one, like using this kind of drones, seems to be very promising.
Excellent. So in your estimation, you're just starting to work on this with the G2 breeding region, I think, is that correct? But what's the potential growth increase with these advanced breeding techniques compared to traditional breeding techniques? How much faster, I guess, are you going to be increasing the growth of these trees, or other attributes, I guess?
So this is, the breeding programs are like a work in between different partners. So everyone is bringing new ideas and new knowledge and work. Like the industry is interested in the gain. The government is always interested in helping and maintaining diversity. And from the university, we are like bringing new ideas. And the use of SNPs is very good because we can get the structure that I showed in the very complicated matrix or very complicated diagram, which is it's gonna be helping both partners to get gain and also to ensure diversity. And that is the idea, to have everyone happy with this technology and see that we can definitely know which parents we are analyzing, but like we don't know which trees we are using to estimate the gains. So with this technology, we have 100% sure the idea which trees we are analyzing with in our calculations.
Okay, excellent. Well, thank you, Esteban. I think we're going to move on to the next presentation here now. But I just want to remind everybody is that we didn't have much time here, but all of our speakers have their own table. So in the next poster presentation, if you want to follow up with Esteban or any of the other speakers, they will be at a table, and you can find them in our next poster session. I believe we're on floor 2. Is that correct?
Yes.
Yeah, excellent. So you can find them on floor 2. Okay, thanks, Esteban. Okay, so now we are going to move on to our next presentation, and this is from Kazi Hossain. Kazi is a postdoc in the Silviculture lab working with me and Vic Liefers, and he's going to be talking about modeling intensive silvicultural yield projections and sawlog profiles for conifer plantations.
Hello everyone. I welcome you all to my presentation. Before I begin, I would like to thank Weyerhaeuser Forest Company, particularly Bob Winship, Kerry McKay, and Dave Swindlehurst for providing funding support for this project. I would also like to thank Forestry Corps, especially Ted Gruding and Andrew Johnson for their collaboration on this project. Today, my talk is about modeling intensive silviculture yield projection and sawlog profile for conifer plantations. Now, let me begin with intensive silviculture. Intensive silviculture is a precision silviculture where treatments are matched to sites' best potential and management objectives. It increases individual tree growth and shortens the harvest rotation. For example, with intensive silviculture, we can achieve an MAI of 6 to 8 cubic meter per hectare per year at best sites at a relatively shorter rotation of 50 to 60 year. Few other benefits of intensive silviculture. It improves the stand merchantability and thus makes stand eligible for earlier harvest. By shortening rotation, it also helps in mitigating risk of timber losses. Now, how do we implement intensive silviculture? We do it through plantations and proper density management on the best productive sites. Now, many areas in Alberta are experiencing midterm timber supply shortages. In some cases, overall piece sizes are also in decline. Therefore, Alberta needs to adopt new forestry practices such as intensive silviculture. But Alberta is not there yet to adopt this practice. Not many scientific studies are also available at this moment. Therefore, the goal of this project is to bring more information to light on intensive silviculture forest management. Currently, I am working on developing enhanced forest management yield curves so that I can assess their impact on timber supply models. As a case study, Weyerhaeuser-Pembina Forest Management Agreement Area, or FMA, is selected. In this FMA, pure conifer land base represents a good portion of their active land base. Active land base is about 51%, of which 25% is pure conifer. Therefore, there is a very good potential of intensive silviculture in this FMA. Now let's talk about our modeling approach. We used individual tree level model like MGM for growth and yield projections. MGM is validated in the context of Alberta. Individual tree data from managed PSPs at their performance age were processed through MGM. Different silviculture scenarios were simulated by varying the intensity and timing of thinning on a range of productive sites. However, in this presentation, I am only presenting a comparisons of results between two scenarios in the pure pine strata. One is the control or no thinning treatment, and the other one is the pre-commercial thinning followed by a commercial thinning scenario. Here, both treatments were modeled for established plantations. Pre-commercial thinning was applied at performance age, and commercial thinning was applied at the age of 35. So let's examine the results now. We can see here volume responses in the first graph. In this graph, solid lines are representing thinning scenarios, and anything that are dashed are representing the control or no thinning scenario. Treatments were applied on a range of site conditions such as best, medium, and fair sites. Here, best sites are represented by red circles, medium sites are represented by blue triangles, and brown diamonds are representing fair sites. No treatments were applied on fair sites. So what we can see here is about 1.5 to 2.5 times yield increase from fair to medium to best sites. That means site productivity is the major driver in volume yield. Therefore, we need a better characterization of site productivity in the land base. However, we also can observe a slight reduction in final merchantable volume in intensive silviculture scenario. But let's examine the piece size responses in the second graph below. Here we can see a gain in piece sizes on the best and on the medium sites due to intensive silviculture. For best sites, I'm comparing the solid line, red solid line versus the red dashed lines, and for medium sites, I'm comparing the blue solid line versus the blue dashed lines. So right now, let's compare the stand merchantability between the two scenarios. The top graph is the stand structure of intensively managed scenario at best pine sites at age 50, and the bottom graph scenario at age 80 on similar site. We can clearly see a difference in stand structure between the two scenarios at different ages, age 50 and versus age 80. In intensively managed stands at age 50 are producing about 548 stems per hectare of commercial timber. That means trees that are greater than or equal to 20 centimeter dBH. Moreover, we also see that in these stands, the number of smaller trees that are less than 20 centimeter dBH are substantially less, which is approximately 187 stems per hectare. In comparison, unthinned stands at age 80 are producing about 520 stems per hectare, which is slightly lower than the intensively managed stands. However, these stands are also producing a significant number of smaller sized trees, like approximately 1,700 stems per hectare. And we all know that commercial timber value of these smaller trees are way less. Therefore, by examining these two graphs, we can say that intensively managed stands can meet the merchantability criteria sooner, can provide volume, and also can give us better value. So what is next? The long-term project of this, the long-term goal of this project is to develop an intensive management decision support tool for forest managers. However, in the short term, we need a better characterization or better prediction of site productivity over the landscape. We need to develop ilkers for all other strata, such as spruce and mixed-wood forest types. We need to examine diameter distribution across silvicultural treatments at different felling ages. Furthermore, most importantly, we need timber supply analysis combining both the volume and the value of the stand. And then finally, I want to conclude my presentation on 2 points. First, our initial results are encouraging for Alberta to adopt intensive silviculture, particularly for spruce and pine plantations. And why shouldn't we do it when Alberta has the best soils in the Canadian Boreal? Besides, similar regions like Alberta have shown promising results with intensive silviculture. Second, while this study is ongoing in Weyerhaeuser FMA, I believe the results from this study can be equally applicable to other FMA holders across Alberta. Thank you very much for sharing your time with me.
All right, thank you, Kazi. So there's a question that's come in here on the chat box here, but will engineered lumber products offset the need for larger products? And will this also offset the need for expensive pre-commercial treatments?
Can you say that again?
So you talked about that desire to have larger logs, but will having engineered lumber products offset the need for having larger logs, and perhaps that will reduce the need for expensive pre-commercial thinning treatments? Maybe we won't need to do this if there's not a premium for larger logs in the future. What do you think?
Well, I think we need larger logs for the industry, right? So because the handling, the processing cost of the larger timber, there are other associated benefits, right? With them, it's not about like the one benefit, but there are other benefits of having the larger piece sizes, right? And then we are not only focusing on the larger piece sizes, but we are focusing on having the larger piece sizes in a reduced period of time. So the shorter rotation, right? We are not looking at longer rotation and having the larger piece sizes, but we are having the larger piece sizes in the shortest possible of the rotational time. So it has other associated benefits. it can minimize the risk, as I mentioned, like insect and another risk. It has fire risk. It can minimize those things. So I think we need this practice, and we need to do that. So what was the other part of the question, I think?
Nothing. I think you addressed it quite well there. Thank you. There's one more question that's come up here, but about your simulated thinning regime. So what the thinning from below, and perhaps you can talk a little bit more about what the thinning regime that you applied in these stands was and why you chose that.
So my thinning treatments was thinning from below, and so that we can remove the less productive, less potential trees from the stand, and allocate those growing space to the higher potential trees, so that we can maximize their diameter growth and have the piece sizes earlier in the rotational age. And my thinning treatments were those were really fully stocked stand. So we applied a sequential thinning. The pre-commercial thinning density was 2,000 stems per hectare, and pre-commercial thinning was applied at the performance age around 10 to 14, like that window, and then the stand was subsequently thinned around age 35 with 800 stems per hectare. And the reasons for those thinning regime was because we targeted a goal. We thought, okay, when can we achieve an economic stand? So we targeted a stand that will provide us like greater than 20 centimeter DBH. We targeted about 500 stems per hectare. When can we achieve that economically merchantable stand? So this is why that thinning regime gave us that. But there could be other treatment combinations as well, which this is why I mentioned actually we need a decision support tool, right? We can have different thinning regimes, different. It's always a trade-off, right? How much volume we want to trade off to achieve the bigger piece sizes. So also like we don't want to target too much volume so that we have suboptimal diameter growth. So it's always, this is the balance that we want to find. And it depends on the management objectives.
Yeah, excellent. Okay, thank you very much, Kazi. So I think we're going to wrap it up for questions for you here. But like I said, if you have other questions for Kazi, you can join him at his table during the poster session. So thank you very much, Kazi. So we're going to move on to our third speaker for the afternoon here. That's Shovon Tanvir Ahmed. He's a postdoc in the Ecosystem-Based Forest Management Lab with Dr. Charles Nock. And he's going to be talking about using imagery from unmanned aerial vehicles to analyze variation in snag frequency.
Hi everyone, welcome to the world of unmanned aerial vehicle. Tree mortality rates have increased steadily over time in many regions, leading to growing concern that some of forests may become net carbon sources and trigger damaging positive feedback in the carbon climate system. Massive tree mortality has already been reported in Canada. Forests in Western Canada are more vulnerable to the effects of climate change. There is already a shortage in water availability in this part of the world. It remains a challenge to properly evaluate the strength of these effects because low rates of background canopy mortality. Unmanned aerial vehicles can easily survey areas several hectares in size that encompass several hundred canopy trees. We can get high-resolution data in cheaper price. Here is my study area. I carried out this study in Cypress Hills Interprovincial Park. It's located in the southern Alberta-Saskatchewan border in Western Canada. Here's the landscape of Cypress Hills. The forest dominated by lodgepole pine, white spruce, trembling aspen, and occasionally balsam poplar. White spruce can be found mostly in the wet areas. Lodgepole pine are found on the ridge in the dry areas, and white spruce or trembling aspen are found in between. In this study, I show how aerial imagery from UAVs can be used to study tree mortality processes by examining how snag frequency varies with biotic and abiotic conditions across a forest landscape. There were 46 forest stands. Each stand was 250 meter by 150 meter in size. A Phantom 4 was flown, maintaining greater than 90% side and frontal overlap between images while taking RGB pictures. I used photogrammetric software Agisoft Metashape to generate 3D RGB point clouds and orthophotos. This is an example of an orthophoto. The raw point clouds were normalized to ground height by subtracting values from a digital terrain model from their heights. The normalized point clouds were subsequently used to generate 10 centimeter resolution canopy height models, which represented the height of the upper canopy surface above the ground. Here the red circles are tree canopy and the green ones are the ground. Tree tops were identified in this canopy height models by using a circular moving window 3 meter in diameter to detect local maxima. We then used a region growing algorithm to segment individual tree crowns. There were 44% commission error and 41% omission error while segmenting individual tree crowns. I used 32 structural and spectral variables for each crown segment and used these to classify the segments as live or dead and to species. Random forest model was used to classify the individual crowns. The model provided over 85% accuracy. Here the yellow color is a dead spruce and the blue one are spruce and the purple polygons are trembling aspen. We identified a total of 45,600 crown segments in our UAV survey, of which around 3,000 were classified as dead. In this figure, we can see shorter lodgepole pine trees as well as those located in stands with higher soil moisture were more likely to be dead when surrounded by tall canopy. So in the x-axis we have neighborhood canopy height, and on the y-axis we have probability of snag occurrence. Neighborhood canopy height is I estimated the height of the neighboring trees located within 10 meters of an individual tree. None of the 3 tree species showed much change in snag frequency at different levels of soil moisture. both lodgepole pine and trembling aspen were more likely to be dead in stands with a taller overall mean canopy height. But white spruce showed a slightly opposite trend. White spruce was also more likely to be dead in sites where it represented more than 80% of the total live canopy area. In contrast, trembling aspen were somewhat more likely to be alive in sites where they were the dominant species. So we found interspecific competition might be killing the white spruce, and maybe interspecific facilitation is helping the trembling aspen to be alive. Most of the variation in snag abundance among stands remained unexplained. The model successfully captured differences in the frequency of lodgepole pine and white spruce snags among stands, but did not have much predictive power for trembling aspen. This study was able to identify thousands of individual snags and relate their occurrence and frequency to biotic and abiotic factors. In future, in this lab, I'm going to use UV Lidar. UV Lidar can provide us more information on structure and for better view of the ground. I'm also planning to use multispectral Lidar data to classify hardwood trees because RGB point clouds were not really good at classifying the broadleaves. Unmanned aerial vehicle might not be suitable for our FMA forest inventories. However, they can be used on a smaller area to get some more detailed information. Here I would like to thank University of Regina and University of Alberta for giving me the opportunity to present this study. And I would also like to thank my postdoc and PhD supervisors. This study was done during my PhD. And thank you very much for your attention.
All right, thank you, Shovon. So we've got a question come in here on the chat box, but how did you measure soil moisture in this study?
I did a gravimetric method, like there were two plots selected. One were in open canopy, another was in very closed canopy, dense forest. And in each plot, I had 3 quadrants. So I dig a pit because it was a rocky area. I dug a pit and collected soil from maybe 15 centimeter below the surface. Then I did it 3 times during the summer. So with time, the soil moisture decreased. That's why we modeled soil moisture based on the midsummer. That's how we can take into account the change in soil moisture over time.
Okay, so this is a point measure. Excellent. Okay, we have another question here. What are some of the costs to outfit a drone with LiDAR capabilities?
My boss knows it better than me. You should ask Charles about it.
Okay. Yeah, I don't think it's trivial though, right?
So some of the cost of to outfit a drone.
Do you have any idea or not really?
Maybe $15,000 around that. I really don't have clear idea about that.
Okay, but you're going to be using that for part of your work starting this year, is that correct?
I think Charles, he's better able to answer this question. Yeah.
Okay. So there's another question that's come in here, but what advantages will be provided with LiDAR versus the photogrammetric point cloud approach, and how will this improve your models? So what's the benefit going to be from using Lidar?
Because with RGB, like photogrammetric software, we used the reflected light. Sometimes in forest it's not possible, but Lidar they use the laser, so they have better penetration through the canopy, so it can give better output.
Okay, excellent. All right, well, thank you very much, Shovon. So we'll kind of wrap up our presentation part of our event today. But like I've mentioned before, if you want to follow up with any of our speakers, they do have a table on floor 2. You can see them then. I'd also like to mention that we do have a small token of appreciation for all of our speakers. So this is a Forest Industry Lecture Series mug and notebook. It's written like it's inverted because it's on through the camera. But anyways, we'll be getting these to you guys in, well, whenever we're back on campus and can see people again. So hopefully in the next little while, we'll get these to each of our speakers. So thank you very much to all of our speakers. And now we're going to turn it back to the next poster session, okay? So a couple of reminders. The posters are on floors 1, 2, and 3. If you can't see where the posters are, you might have to drag the screen up. I know there were some issues with that in that first session, but you can hold the mouse button down, you can drag the screen up, and then you can see the bottom row. Sometimes, depending on the sizing of your screen, you can't necessarily see all the tables, but they're all in the bottom rows of each of the floors. So that's where the posters are. Another reminder, please enter your votes for the poster prizes. We'll be tabulating those at just before 5 o'clock, so get your votes in. There's the link to the voting for the poster prize has just been posted in the chat box, so you can get it there. But with that, we're going to go back to our poster presentation, and we will wrap up here just before 5 o'clock. We'll reconvene back in the plenary mode for the wrap up. Okay, so enjoy the rest of the time of the posters. All right, well, welcome back everybody. Hopefully you had a chance to visit some more of the posters and talk to some more of the students. I just wanted to do a quick wrap up here at the end here, but I just wanted to thank you all for attending today. And I have a couple other thanks to for the people who make this happen. So in particular to Byron Flores, Rahana Bennett, and Christy Nohos. Without them, this event really wouldn't have happened. So a big thank you to those folks. I also want to announce our poster winner. So thank you to everybody who voted. We did end up getting a lot of votes in this, which is great to see. People were engaged with it. But our 3 winners are Gigi Baker, Gleb Kravchinsky, and Nick Bakilo. So each of you will be receiving a $50 gift card to the bookstore, and Christy Nohos has those, and we'll be in touch with you soon about how to collect those. So congratulations to our 3 poster prize winners. Way to go. Last thing I want to do is announce our next Phil's lecture. So the 86th Forest Industry Lecture Series is scheduled for Thursday, November 4th. We have Dr. Terry Sharrick from Michigan Tech University, and he's going to be talking about forestry and natural resource education. We originally had Terry planned to come last fall. Obviously, that didn't happen. So we're really glad that he's going to be able to join us for the next one. And actually, the timing worked out pretty good because he's in the midst of doing a big review of forestry and natural resource education across North America. So he'll be able to give us all the latest and greatest next November. So please mark that on your calendar. Hopefully we'll be able to be an in-person event. We'll be able to have an actual lecture and hopefully a dinner after at the University Club. If all goes according to plan, we should be able to do that by November, I really hope. So once again, thank you all for coming today. We will have this remote platform open for another half an hour, so you can see the countdown timer is started up in the top left-hand side there. So we'll be open if you want to stay around and chat. Remo will be open to do that for the next little while. But other than that, thank you all for attending, and we'll see you all again at Phil's in November. Bye for now.